Hello, my name is Charles Blatti, and I want to give you a quick overview of our new visual analytics tool for tumor phylogenies, PhyloDiver. So the significance of our new tool is that it is in the domain of cancer phylogenetics, which is the study of the progression of mutations in tumor cells. Cancer phylogenetics works by trying to reconstruct the evolutionary tree that describes the different subclones in a tumor population. In the purpose of reconstructing that tree, we hope to better identify individualized targets for treatments of an individual, to better understand the cancer's progression, and then to discover patterns of evolution across many samples. And so the goal of this particular project is to help broaden the impact of cancer phylogenetic workflows with improved visualization tools for researchers. So standard visualization tools typically fall in one of two camps, sort of static images like this fish plot shown here, which can spatially capture the cellular prevalences of different subclones in the samples, but really do not allow for dynamic investigation. The other type of visualization are interactive tree visualizations. They allow for the selection and exploration of the evolutionary tree, but they're general purpose, and so they don't really have a mechanism for spatially conveying information about the subclone proportions. So that brings us to PhyloDiver and some of its key unique features. First is the feature of dynamic proportions. So PhyloDiver shows in its main view the evolutionary tree, and for a selected sample, the cellular proportions of the subclones. And the cellular proportions are informing the heights of the nodes, the leaves of this tree, and this dynamically updates for the user. So if they select a different sample, if they select a particular mutation cluster, or they select a particular subclone, all of these proportional heights will update throughout the main window. The next major feature of PhyloDiver is its integration with different annotations. So when you select on a mutation cluster edge, you get a summary of information about that mutation cluster. This summary includes what are the high impact mutations according to the variant effect predictor. Of those mutations that occur in genes, which of these genes are known cancer genes from the cancer gene census, and then also which of the unique genes that the mutations fall in have known drug gene interactions from the GDI database. And besides seeing this summary information on the annotations, you can click on a row of the summary table. So here, severity equals moderate, to get even more detail from the original VCF and annotations. So this drives the user to all these underlying details that match this particular mutation cluster and the severity impact of moderate that we had selected. And then you can still use the tree navigation to jump to different subsets of tables, either for a mutation cluster or for a subclone. So those are the major new features in PhyloDiver. Users, if they wish, can upload their own data into PhyloDiver. We have a server available at phylodiver.org where you can upload your own data. And the data needs to follow a specific format that is defined in our specifications on the website. So it's a JSON format. And another way that you can produce these JSON files other than reformatting the data yourself is to run our standardized fair workflow, PhyloFlow. So PhyloFlow takes VCF files, runs them through the different steps of cancer phylogenetic analysis, and then finally merges all these different outputs from each step into the JSON file that PhyloDiver expects. And when you upload your data, you're uploading it locally and exploring it on the client side of your browser. And in the future, we would like to take this interactive interface that we've created and embed it in self-contained HTML files or in Jupyter notebooks. So when users run the Dockerized PhyloFlow workflow, it will automatically generate those outputs, and then those outputs themselves can be viewed within different genomics cloud platforms, such as Terra, besides being viewed on your local machine. I'd like to thank the Cancer Center in Illinois for funding part of this project, and all my collaborators from UIUC and from Mayo Clinic. And if you have any questions, please contact me at blatti@illinois.edu. Thank you.